So let me share my recent work. The title is Succinic semialdehyde derived from the gut microbiota can promote the proliferation of adult T-cell leukemia and lymphoma cells. ATL, the disease targeted in this study, is a T-cell lymphoma caused by a virus, HTLV1. The development of ATL occurs when infected cells turns to monoclonal proliferation after infection, HTLV1 infection. The onset of the disease caused severe immunodeficiency and so on. It is serious disease with a very low survival rate even after chemical treatment. However, it is also characteristic of the disease that not all infected individuals develop ATL, with only about 5% of infected individuals developing ATL after approximately 70 years. HTLV1 infected persons who do not develop ATL are called HTLV1 carriers. The triggering factor for the development of ATL remains unknown. One suspected trigger has been identified as frequent mutation to the NF-kappa beta pathway, metabolic pathway responsible for the inflammation. As you know, there are known causes where substances derived from gut bacteria can aggravate blood cancers. The figure showed the association between acute myeloid leukemia and gut microbiota. Disruption or dysbiosis of the gut microbiota leads to proliferation of AM cells and progression of AML. Based on this background, this study hypothesized a link between ATL and gut microbiota. To test this hypothesis, I set two purposes of this study. The first one is gut microbial alteration in ATL or future patients. The second one is specific metabolites affect infected cells, HTV infected cells to proliferation. The number of samples used, 2 cohorts were used in this study. Only cohort 1 had carriers, and of which 8 were high-risk carriers. And the cohort 2 also included more advanced ATL patients than cohort 1. Now I come to the result. First, two-sided comparison of bacterial abundance was performed to identify bacterial associated with the development and the progression of ATL. The groups used to comparison were healthy control and low-risk carriers combined, and ATL and high-risk carriers combined. As a result, 7 bacteria were identified that significantly increased in ATL and high-risk carriers. And these 2 bacteria are possibly correlated with tumor markers of ATL. And I also performed functional gene profiling profile analysis. In functional analysis of gut microbiota showed that approximately 400 bacterial genes were significantly increased in the gut of ATL patients. And I also performed enrichment metabolic pathway analysis. Then there are several metabolic pathways significantly enriched in ATL patients. And in this study, I focused on one pathway. This is homoprotocatechate degradation pathway, because this pathway was found to produce succinic semialdehyde called SSA, and this SSA is known as oxidative carbonyl, and this effect provide oxidative stress, and oxidative stress reported to be involved in carcinogenesis. So, and it also inferred that Klebsiella, which was increased in the ATL patients mentioned earlier, and positively correlated with the tumor marker, is involved in this pathway. So we added this SSA to the oncogenic leukemic cell derived from ATL patient cell line. Then we can see the significantly increased of the ATL cells, especially 200 micromolar SSA addition. Summary, in this study, I hypothesized that firstly, gut microviral alteration in the ATL or future patients. The answer was Klebsiella and the other candidate 6 bacteria. And second one, specific metabolites affect the infected cell proliferation. The answer was succinic semialdehyde, SSA. That's all, and this is the summary figure. Thank you.